Hey everyone, so today in this video I'm gonna give an overview about apoptosis. Basically, apoptosis is the programmed cell death. The cell activates the enzyme that degrade first the nuclear DNA and second the nuclear and cytoplasmic proteins. So basically in the apoptosis, plasma membrane remain intact and apoptotic cell break off, giving apoptotic bodies, which is phagocytosed by the macrophages. Apoptosis does not elicit any inflammatory reactions because the plasma membrane is intact. So let's there's a normal cell. Now there is a cell injury or any trigger that will lead to the some activation of proteins and also some morphological changes like condensation of chromatin, formation of blebs, there will be cell shrinkage, which will lead to the formation of apoptotic bodies, and hence these bodies will get phagocytosed by the macrophages. Now coming to the topic of causes of apoptosis. Basically apoptosis occur in two broad contexts. So basically as a part of normal physiological process and also as a pathophysiologic condition. Now in the physiological condition, apoptosis occurs when there is a need of removal of excess of the cells during embryogenesis or during development, or it could occur in the involution of hormone dependent tissues on hormone withdrawal. For example, endometrial cells break off during the menstrual cycle. Apoptosis occur when there is a cell turnover in the proliferating cell population, such as immature lymphocytes in bone marrow and thymus, or in the case of death of host cells that have served their useful purpose. For example, the neutrophils in an acute inflammatory response, after they have served their purpose, they will get into the process of apoptosis. Now coming to the pathological causes. So apoptosis eliminates the cells that are injured beyond repair. So if there is a DNA damage which could occur by radiation or cytotoxic anti-cancer drug, so or the accumulation of the misfolded protein, so cell death triggered by the improperly folded intracellular protein. Apoptosis also induced during certain infections, mostly during the viral infection. Now coming to the changes that occurs morphological. So what are the morphological changes that occur during the apoptosis? So first there will be a cell shrinkage. So cell size is reduced and cytoplasm becomes dense and eosinophilic. It becomes eosinophilic because the proteins are not tightly packed, and there would be chromatin condensation, which is one of the most characteristic feature of apoptosis. There will be formation of apoptotic bodies and the cytoplasmic blebs. And the last process, that is a phagocytosis, which generally occurs by the macrophage. So basically now coming to the mechanism underlying the apoptosis. So basically in apoptosis results from the activation of enzyme called caspases. But there are two phases in the apoptosis. The first phase is your initiation phase and second is your execution phase. So there is a caspase. Some caspase gets activated by the initiation phase, and now these activated caspase will further trigger the terminal caspases, which will lead to the cellular fragmentation. Now coming to the two pathways of apoptosis. Before getting into this, let's see what are the anti-apoptotic proteins and your pro-apoptotic proteins. So basically in the cells there are anti-apoptotic proteins and pro-apoptotic proteins. So basically anti-apoptotic protein like BCL2, BCLX, MCL1. So these proteins reside in the outer mitochondrial membrane as well as in cytoplasm, and they keep the mitochondrial outer membrane impermeable. They prevent the leakage of cytochrome C and other death inducing proteins. Now coming to the pro-apoptotic protein like bags and bag. So on activation, these oligomerizes with the outer mitochondrial membrane and enhance its permeability. Now this will lead to the leakage of cytochrome C and other proteins, which lead to the activation of caspases. Now let's come into the two pathways of apoptosis. So basically the first is the mitochondrial pathway. But let's see what happens in a viable cell. When there is no cell injury, there are some the survival signal like growth factors that attach to the receptor, which lead to the production of anti-apoptotic proteins, which will not lead to the leakage of cytochrome C. But when there is a cell injury like the growth factor withdrawal, DNA damage, protein misfolding, this will lead to the activation of sensor like DH3. Now this will activate the pro-apoptotic proteins. There will be leakage of cytochrome C and other proteins, and there will be the activation of caspases. Now once released into the cytosol, the cytochrome C binds to a protein called APAF1, that is your apoptosis activating factor 1, form a multimeric structure called the apoptosome. Now this complex binds to the caspase 9. Now this active caspase 9 then triggers a cascade of caspase activation by cleaving and thereby activating other pro-caspases. So basically by the mitochondrial or intrinsic pathway there is activation of caspase 9. Now coming to the death receptor extrinsic pathway. Now this pathway is initiated by engagement of plasma membrane death proteins. So this is also known as receptor mediated pathway. These include those activated by the death ligands. So death receptors are the cell surface receptors that transmit apoptotic signal, which are initiated by specific ligands. Now these receptors activate the death caspases. Now death receptor belong to a superfamily of TNFR, that is your tumor necrosis factor receptor, which are characterized by cysteine rich extracellular domain. Now adapter molecules like fast associated via that domain or tumor necrosis factor receptor 1 associated that domain, they contain death domain so that they can interact with the death receptor and transmit the apoptotic signal. The best characterized death receptor are FAS and TNFR1. Now this FAS ligand, which is a homotrimeric protein, act as a ligand for FAS and cause oligomerization of its receptor upon binding. Now the complex of FAS, FADD and procaspase 8 is called death inducing signaling complex. Now this active caspase 8 then activates the downstream caspases, that is caspase 3 and 7, committing the cells to the apoptosis. So basically the death receptor or extrinsic pathway ultimately leads to the activation of caspase 8. So these two pathways occur as an initiation phase of apoptosis. Now coming to the execution phase of apoptosis. So this is the final phase of apoptosis. Now the active forms of these caspases like caspase 9, caspase 8 and caspase 10 trigger the rapid and sequential activation of executioner caspase, that is your caspase 3 and caspase 6, which act on these cellular components. So they will lead to the endonuclease activation, which will lead to the DNA fragmentation. They will lead to the breakdown of cytoskeleton, that will lead to the formation of cytoplasmic blebs. Now coming to the last topic, that is a removal of dead cell. So how these macrophages know that these bodies need to be phagocytosed? So in the healthy cell, the phosphatidylserine, this lies on the inner leaflet of plasma membrane, but in the apoptotic cells this phospholipid flips out and is expressed on the outer layer. Now this is recognized by the macrophage receptor. Also, apoptotic bodies may become coated with natural antibodies and proteins of complement system, which are recognized by the phagocytose. This process of apoptotic cell phagocytosis is known as apoptosis, which is so efficient that dead cells disappear often within minutes without leaving a trace. So this is all about the apoptosis, and I hope this video helped you in some way, and if you enjoyed this video, please do like, share and subscribe.